everyone. My name is Akila. This talk is about scaling genomic workflows for efficient genome analysis. I'd like to acknowledge Dr. Jason Pitt, who has been leading this effort at Pitt's lab in CSI. So at Pitt Lab, we use computational approaches to understand multi-omic genome instability patterns in cancer. This requires a lot of data. So one of our side projects is to build and use scalable workflows for processing genomes from cancer patients. With the resulting varying calls, we use novel AI approaches to get a unique perspective on multi-omic patterns. Then we leverage this to enhance our understanding of differences in mutational processes across cancers and the implication of those patterns for precision oncology. So running bioinformatic tools on massive amount of data being generated today using next-gen sequencing technology is very expensive. Therefore, it is important to understand the underlying cost of operating a large-scale infrastructure platform. So when you try to scale biotools vertically, you realize that they cannot effectively utilize additional computing resources to increase throughput. So we need a better framework. So Swag is a software framework that we have developed using Parsl that is specifically tailored towards running genome analysis workflows at large scale. It is portable across various execution platforms like Aspire 2 supercomputer or AWS Cloud. So you can create a workflow configuration with all the input files in BAM or FASTQ formats, and you can include additional metadata, so special cases like group IDs, and whole genomic sequencing or even whole exome sequencing sample metadata. And we heavily rely on Parsl to parallelize the workflow and achieve high resiliency and full tolerance capabilities. So here's a snapshot of our complete workflow. So basically there are two pipelines. So one is Genomic Data Commons, which provides the cancer research community with a unified data repository with tens of thousands of sequenced cancer samples. So all the samples are processed using the same pipeline, which has been comprehensively documented and publicly shared. However, they do not provide a scalable workflow that would allow researchers to process DNA sequencing samples at scale. So we have replicated that same DDC pipeline using Swag, which again uses Parsl for its parallelization capabilities. So this is basically our attempt to solve that problem of processing petabyte scale genomic data. So for every BAM file that we process for each patient, we split that into individual contigs, basically multiple chunks based on specific regions in the human genome. So each of these contigs can be processed individually, so we scale out depending on the number of chunks that we have created. So we have managed to optimize this workflow by creating multiple executors. So huge shout out to Ben, who has been helping us with Parsl configuration. So basically we have a general executor that can run pretty much IO bound tasks. And we have another compute optimized executor with a higher workload to CPU ratio, so that can run compute intensive tasks. And we also have a couple of long running tasks. So those ones are assigned to a special executor that can make use of specific queues on Aspire 2A that can run long running tasks. So Aspire 2A is actually the Singapore's national petabyte scale supercomputer that we usually use for running our analysis. So up to this date, we have processed more than 12,000 patient data files amounting to petabytes of data, and we have produced some scientific work out of that. So this is our roadmap to basically build a multi-site genomic workflow. So we are planning to make use of AWS, specifically Amazon EKS, Kubernetes API to basically submit jobs over Parsl. And we are planning to use Amazon EFS to create a shared file system so that tasks can easily access inputs and share its outputs with the downstream tasks. So here are some opportunities to improve our pipeline. So as I mentioned earlier, we split genomic data files into multiple chunks, and some of these chunks can be high in size because of biological variability. And we have noticed that bigger chunks run into timeout issues. Sometimes Parsl will spin up more instances than it needs to due to load balancing, but this can be very costly, especially when running the workflow on a cloud platform like AWS. So we can address some of these problems to a certain extent by improving Parsl capabilities. For example, duration-sensitive task allocation by making use of user-provided hints that you can pass to the scheduler so that it would allocate tasks depending on how much time left to execute that on a specific node. Also implementing a cost-effective task packing strategy so that Parsl will use minimum number of nodes possible for specific queues, especially when you try to run things on AWS. Yeah, that's it from me. Thanks, everyone.